There will be another Socratic activity at the end of class where you'll do more relevant questions. So before we get started, this study in the Proceedings of the National Academy of Sciences, the prestigious journal, the United States of America, came out a couple of days ago, and it really hit close to home. So I wanted to point out a couple of things. In course evaluations I get in this class, and in all the classes I teach, there's always a small but significant number of students that say, I hate the class format, it doesn't work for me, and I understand why, especially because a lot of students, by the time you've gotten this far, have figured out how to succeed in lecture-style classes where you come in, you take notes, you get tested in multiple choice, true, false, fill in the blank, labeling, maybe some short answer. But it turns out that active learning, that is this process where we do things in class together and you access the material outside of class, actually works better for scores. So this was particularly interesting to me because this study says students rated the quality of instruction in passive lectures, like when you just sit there and take notes and I talk at you for 50 minutes, more highly. This was physics students, not biology students, but still. Students rated the quality of instruction higher. They have a preference to have all of their physics classes taught this way, and then the punchline. Even though their scores on independent tests of learning were lower than those in actively taught classrooms. In other words, students, I'm not saying you do, but in this study of this class they taught, the students said, yeah, we like just sitting here and listening better, but they also did worse on tests. That's why we do active learning, is because it's proven, depending on how well I execute it, to be better at improving student grades and understanding of the material. So the bottom line is, if you feel like a lot of those students that would rather me just lecture and not have to watch videos before class, and I just sit up here and draw on the board and give you facts to remember, I understand where you're coming from, but the evidence says that this type of approach is more effective. So I'm hoping you'll be on board with me for the rest of the semester in this approach. There's evidence in literature. Yeah. And so you may feel some of these feelings when we do small group work or larger group work as we go on in the semester. Instructors that gave up on this sort of approach, the active learning approach, reported things, you may be familiar with some of these, like students dislike being forced to interact with one another. Group work, right? I didn't like group work either when I was a student. I still kind of don't as a faculty member. That is group work with my peers, not with you. And they resent the increase in responsibility for their own learning, despite, of course, the fact that once you graduate, you are entirely responsible for your learning for the rest of your lives, so you might as well get used to it now. So again, that's just perspective. That's where I'm coming from as a teacher. We're still working on genomes, so hopefully we'll wrap up genomes today. We'll see how we do. And I want to return to this question because I kind of skipped over it last time. By the end of class, we'll talk about individual human-to-human genetic variation in genomes, not just human chimp, human bonobo, which we talked about a bit last time. So one of the exercises that I'd asked you to work on for today was this. Find the sequence of the breast cancer susceptibility gene. Know its GenBank accession number. If you don't know what that is, there was a video that talks about using GenBank and accession numbers, but we can answer questions about that if you have them now. Find out how many times this particular sequence occurs in the sequence of this gene, and then calculate how often we expect that sequence to occur. Did anybody try this and have any questions about it? I've dedicated significant time in class to either answering your questions or working through it with you together. We can do either of those things. Yeah? Okay. Good. So you found it on another website, didn't know how to find it on PubMed. That's fine because PubMed, which I didn't tell you, should have, so thanks for bringing it up. PubMed is for searching literature, specifically journal articles. So there's another database at NCBI, which may be the website you found, that's good for looking up genes and gene sequences. Did anybody find the breast cancer gene sequence? Because if not, I've provided it for you. Just wanted to know if anybody had any questions about how to find it. So what I would do, since no one's talking, I'm going to go through this until somebody tells me that they already did it, and then I'll stop. So in web browser, we'll go to the NCBI website. ncbi.nlm.nih.gov. Or you can just Google NCBI, National Center for Biotechnology Information. And you'll see there's this drop-down menu that says searching all databases. That's fine. That's what you can do. If you type in, what's our search term? Trying to find the gene sequence of BRCA1. BRCA1. Then hopefully it will come up with some hits for us. Okay. Top hit. So this is promising looking to me so far. It says gene. Yes. BRCA1 gene. And what I would suggest you do, if you're on this page or if you're looking for the gene, is probably go to that download button. Let me try that too. This doesn't work as well in a browser. So it pops up. Do I want to download transcripts, proteins, or gene? I want gene, because we're talking about genes. We'll talk about transcripts and proteins later, if you're not familiar with that terminology yet. And that will presumably, oh look.
Nice.
There it is. Fantastic. So, things to note. So what this will do if you're working on a laptop is it will probably download a text file to your desktop or something like that, or it will just display in your browser window. So here's the DNA sequence of the Homo sapiens up there at the top. BRCA1 gene. It also tells you what chromosome it's on, except I drew over the chromosome number. So this is the full DNA sequence. The T's and G's, single line code. This is not double-stranded, this is single-stranded, because you can always figure out what the other strand's sequence is by base pair complementation. What format of DNA sequence is this? The text version. We've only talked about one. It starts with the whatever, greater than or less than symbol. This is FASTA. This is a FASTA formatted DNA sequence, which means the first line of the file starts with that symbol, and this is all just descriptive. Obviously, this is not part of the gene sequence, I hope. Teachers are usually told not to say obviously to their classes because obviously not everybody usually knows what we're talking about. But those are letters other than A's, T's, G's, and C's. That's not part of the gene sequence. It starts here at T. TGT, GTG, etc. So this is a FASTA formatted sequence. First line is all descriptive. Just tells you about the gene. The rest of it is the DNA sequence. Any questions? NCBI website, search field, type in the name of the gene you're looking for. If it gives you that download option, you can get the FASTA sequence. So what I've done, you may have noticed, is I sent out a Google Classroom assignment with a file attached, and that is the text file that contains this gene sequence. So you've got that now in case you were having trouble downloading it just now. It's easier to work with if it's in Google Docs, which that attachment that you now have is. So what I'm going to do now is for answering these questions, we've got the sequence of the BRCA gene. We'll talk about the accession number and what that means, and then do these two exercises in Google Classrooms in that Google Docs file. So I'm going to stop recording now because I'm going to switch out of my recording app into Google Docs. So if you're looking for GAATTC, does anybody know what the answer is? Expected. How many different nucleotides are there in DNA? Different types. G's, A's, T's, and C's.
25% for each.
So if you picked one spot in a chromosome, we already talked about this briefly once in class. This is just the full-on version. Yeah, your probability, if you closed your eyes and you had a big piece of DNA sequence in front of you and you poked your finger on you'd have a 25% chance that your finger lands on a G. You've got a 1 in 4 chance that any particular letter you look at is going to be a G. It's like you've got a 50% chance of flipping a coin and getting heads or tails. Does anybody have any concerns with this? Because you should. It would be totally reasonable to raise your hand and say, that's not right. There's some assumptions, in other words, that are made in this. So if everyone is okay, that's fine, I'll move on. But
aren't there parts of DNA that are more concentrated in certain nucleotides than others?
like what vocabulary term that we learned in class earlier this week? Isochores. We know that different parts of chromosomes have more or fewer G's and C's or A's and T's. That is the big assumption here, is we're assuming that the DNA that we're looking at has 25% A, 25% G, 25% C, and 25% T. That's fine. That's an assumption that you should make. If you ever get to this on something that you're stressed out about, like a test, and you're not sure if you should make that assumption, go with it. Say, if you ever, by the way, totally unrelated to this, if you're ever doing something for an assignment or an exam and you're not sure that you're answering it quite the right way because you're not sure what assumption to make, write down whatever assumptions you are making and then answer the question. And I'll take whatever you say you were thinking of into account when I grade it. So if you want to say, this is the way I'm going to solve it, I'm assuming that there are equal numbers of G's, A's, T's, and C's in this part of the genome, that's good. Let's solve it that way. So G's, 1 in 4 chance. What about chance at this spot of finding an A?
25%.
Also 1 in 4. It's like flipping another coin, right? They're independent of each other. If I just closed my eyes and blindly pointed and wanted to know what the nucleotide was there, the probability of that being an A is also 25%, 1 in 4. Could be an A, could be a T, could be a G, could be a C. So the only thing you need to know, you can remember that part, right? Probability of finding any one nucleotide at any one spot is 25%. If you can remember that, then you only have to remember one other thing to do this calculation. The probability of finding any one of the 4 nucleotides at each spot in the genome is 1/4. Then you just need to know what numerical operator to put in between all those 1/4s. What do you do to them? Do you add them together or do you multiply them? Turns out you just have to remember you have to multiply independent probabilities. If you added them all together, what would you get? 1 in, what's 1/4 plus 1/4 plus 1/4 plus 1/4 plus 1/4? 6/4, which is, yeah, 1.5 if added. That doesn't make any sense because you can't get probabilities greater than 1, because 1 means 100% chance of something. You can't get anything bigger than that, so we can already rule out adding. It has to be multiplying. So what happens when you multiply 1/4 by the number of nucleotides you're looking at, or 1/4 to the 6th? It's 1/4 times 1/4 times 1/4 times 1/4 times 1/4. 1/4 times itself, 6 times. That's a big number or a small number?
Small.
It's a fraction that gets fractionated, and fractionated and fractionated. That's the frequency with which you expect to see 6 nucleotides in a row in the genome. What is that number?
You want percent decimal?
Either.
So it's 0.02% chance.
0.02%, which is 0.0002 in frequency. Okay, so what that means is if you looked at any random 6 nucleotides of DNA in the genome, you've got a 0.02% chance of finding that 6 nucleotide sequence. It's really rare, which it's relatively rare anyway. And if you take 0.002, you divide 1 by 0.002, that gives you how many nucleotides, how many sets of 6 nucleotides you would have to look at before you found one of these. So it's not its frequency. The inverse of the frequency is the probability. And what's that? It's 4,096. In other words, so just showing you two different ways to approach this. You can either think about it as you've got a 0.02% chance of finding this if you look at any 6 nucleotides, or you have to look at 4,096 different sets of 6 nucleotides before you'd find one that is that specific 6. So every 4,096 groups of 6, you should, on average, find GAATTC. We're almost there, and then we're going to move on to something else. The question was, how many times in this gene, we know it was, how many times in there? 12. 12? How many times should we see it if this is true? If our expectation is true and it's random nucleotides, we should see GAATTC in it. There's only one more.
4,096 divided by.
It's something having to do with the 4,096, but we need to know one other piece of information.
How many sets of 6 nucleotides were there originally?
How long, right, so which is, in other words, basically how long was the gene? How many nucleotides are in the gene? So if you've got that open still, what does it say in the header of the
81,189.
81,189 base pairs, which is, we could argue about this later. It's basically the same number of multiples of 6 that you have. It's actually 5 less than that, but that's a whole other story. So if you divide that by 4,096, there's a number of times, every 4,096 base pairs, we should see one of these. So if there are 81,000 base pairs, we should see.
I thought 4,096 was how many groups of 6 you have to look at.
It is. We'll talk later.
Oh.
It's because every nucleotide in the gene starts a group of 6. So the length of the gene is essentially the number of 6 nucleotide sequences it has, minus 5. Don't worry about that if you don't want to. Because it makes this math basically the same. That small change doesn't matter. What's 81,000 divided by 4,000? I got about 20. Does that sound reasonable? 4,000 times 20 would be, if we're estimating, if this was 4,000, we multiplied it by 20, that'd be about 80,000. So that's right. So we should see this sequence, that's the answer, about 20 times. If you want to round up or round down, I don't mind. There is no right answer. Maybe it's 21, maybe it's 20, maybe it's 19 and a half. It's, call it 20. You can round up to 21 if you want. So importantly, what does this mean?
I just had a question on.
Go for it.
Lost on the number 4,096.
Let's review again. Yeah, absolutely. So that was. That calculation, the inverse of the frequency, 0.0002 is the frequency with which we see those GAATTCs. So if you take the inverse of that, that's how many times you would have to look at 6 nucleotides before you would find one. So again, you can either think about this as a frequency. You look at one spot, you've got a 0.02% chance of finding that sequence. Or if you take the inverse of that, you find one event every 4,096 times you look. This is going to be a useful skill to do later in the class. The relevance isn't clear now. I get it. But we'll use this again later, and it makes sense to cover this when we're talking about chromosome structure because we're talking about nucleotides and frequencies. Was this the number of, did we observe 20 sites of GAATTC in this gene? We saw fewer, we saw 12. So maybe that means that our assumption's wrong. Maybe it does mean that we're looking in a region of a genome that has fewer A's and T's. So maybe if we corrected for that, we would get something closer to the expected frequency. Not super important. But the point is now you know how to do something to calculate an expectation and how you know to find the observed number of restriction sites in a sequence. Very important for genetics as we go through this semester to be able to do this. And there will be more practice coming as well. Any other questions, concerns? Yeah.
So to interpret this, does that mean if you have that exact sequence of 81,000 nucleotides, you're more susceptible to breast cancer? Or
So relevance to breast cancer in particular, none. Thank you for asking. Let me explain. The reason I chose the BRCA1 gene for this exercise was just because it's a gene that we talk about a lot in the semester, but there is no relevance or no relationship between this 6 nucleotide sequence and cancer risk. So I chose it because it's a well-known gene, not because this sequence is particularly relevant. 12 times. So if we took this gene and we put it into a bacterium and we had that bacterium express whichever restriction enzyme, I think it's either EcoR1 or HindD3 that recognizes this sequence, it would cut that gene up into, if there are 12 sites where that enzyme would digest this gene, then we'd wind up with 13 pieces of DNA. Some people want to know about that. We'll talk about relevance later. That's the main reason people do these sorts of calculations. They want to know if you've got a piece of DNA and you're going to do some cloning, if you're going to make a transgenic organism, if you wanted to move this gene into another organism, for example, and you wanted to just cut out a little bit of it, you'd want to know where these restriction sites are, how many of them there are. Quick one for you. Take about a minute or two, just one quick practice writing a consensus sequence. We did this last time. I just wanted to give you another shot at it. Make sure your answer agrees with your neighbor. Shouldn't take very long, but go for it. While you're doing this, you should definitely come up with a question to ask once you're done. Thanks for saying that, because we'll do that. It's really straightforward, but it's important to go over.
I looked up some videos that says you just put like a -10, and then he showed this long gene sequence thing, and then he didn't really explain how he got to that part.
That'll be more relevant when we get to central dogma and transcription. Transcription's where you get into the negative numbering, but we're not there yet, unfortunately. Okay. Consensus sequence. Coming up with the, what?
It's a big question.
There's a big question.
Yeah, what do you do since there's 2 G's and 2 C's?
Perfect. So the consensus sequence is the average, right? Whatever is most common. So the first site, there's 3 out of 4 that are A, so the consensus sequence is A. The rest of it's pretty straightforward. There are 3 out of 4 T's, 4 C's, 4 G's, 4 A's, and 3 out of 4 A's. What do you do at this second site? Oh, speaking of numbering, these alignments are numbered from left to right from 1 to whatever. So for numbering, in this case, and we'll talk about differences as we go through the semester, this is just 1 through 7 left to right. So you would talk about nucleotide position 1, then we would all know we were talking about the same spot of the alignment. It's just for nomenclature, it's just for communication. You could say, I'm talking about the 4023rd nucleotide, you'd be able to find it by counting from 1 to 4023. So what do you think we should do to fill this in?
Since it's 50-50, you just go C/G.
Okay, so one option, it's 50-50, so you could do, if I was writing with a pen, you could do C/G. That's an option. Half one, half the other.
Whichever one was from the, how would I say it? The oldest organism.
Well, yeah, if you were,
Not like the oldest, like birthday wise, but the oldest, like,
Evolutionarily?
evolutionarily, yes.
Yeah, so if you, once you get into evolutionary biology, you'll talk a lot about inferring which is the evolutionarily oldest sequence. But here, basically, the point is there is no consensus. There's no one nucleotide that's in the majority. So you can either do that, or there are two other options. One, you can write an N if you want to. That stands for any nucleotide. You can't use A because A already stands for A, so you couldn't call it A for any, so it's N for any. The other option, which is something we haven't talked about yet, but you might want to know about more as trivia than something you need to know about, is there are also, we have the 4 letters for the 4 nucleotides. Geneticists have also come up with single letter codes for every possible combination of nucleotides, in which C or G is called S. So you could put an S instead of that C/G. In this case, S stands for strong, and that's because C and G nucleotides make 3 base pairs, or 3 hydrogen bonds between them instead of 2. So if you have someplace that's C and G, you could label that S if you're not sure, if there's not a consensus, but it's either C or G. Guess what A and T single letter code is? W for weak. Okay.
Also stands for weenies,
Or weenies?
because they're weak.
Yeah, exactly. Weak. W. Good. So that's the basics and one exception in how to create a consensus sequence and number it. I guess last, maybe we should cover the N for any really would be best if you had a situation where you had, in this case, a G and an A and a T and a C. There really was there was clearly no consensus. It wasn't like one or the other. It was, there is the option of having any nucleotide at that position. That's really what the N is for. If you actually observe, there is literally any nucleotide or every nucleotide at that locus. So back to NCBI, what was the gene associated with NM_057410.4? That's the accession number. If you go to PubMed or the NCBI database, you plug that into the search field, what do you get? Try it if you haven't. I want to make sure that you know how to do this. And if you don't know how to do it and you don't want to try it on your own, come talk to me and we'll do it together. But it should come up with a record. The top hit should be Drosophila melanogaster, which is fruit fly. And it's, the gene name is EGFR, which stands for, not for bless you, but yes, bless you. Stands for epidermal growth factor receptor. Again, a random gene that I chose just for the purpose of choosing a random gene. Just practicing you getting DNA sequence information and information about genes from reliable online sources. Speaking of which, thank you all, I was really impressed with your first responses, the comments that you left on this question on Canvas. Really good. Really impressive. I haven't had a class that's been this information literate yet, and I've been here 7 years, which is not really a long time, but I've taught this class a number of times. You're doing really well. There's a little bit of feedback I want to give you, but then by the end of class, I want to wrap up with, let's take a vote on this fact. Is it or is it not? So you all found individually a bunch of different references that can be used to support or refute that statement. We share 99% of our genes with chimpanzees and bonobos. So, brief summary of a lot of the comments I saw. A lot of you said, well, if we're going to debate is this fact, is the number itself supposed to be exact? Does it have to be 99 or could it be 98.6 rounded up to 99? Sort of semantics. Like, technically, unless you can find a citation that says that exact thing, then maybe you can't call it a fact. Some of you, this is personal. That is, this is not a geneticist talking on behalf of all geneticists. This is just a scientific writing perspective. A lot of you said things like, I feel this is true because, or I believe something. Up to you. Scientific language typically strays away from the you writing as though you're making a judgment call based on feeling. You'd want to avoid I feel, I believe. Just be passive voice. The evidence shows that, or this citation says that. Therefore, this fact is supported or not. Again, that's really the least important thing I can say from now until the rest of class. More importantly, a few of you, but not very many of you, and this is why I was really impressed, only a few of you posted non-research citations. That was citations to like CNN, a story about the papers that talked about human genome and chimpanzee genome. So this is one example of, it's from the journal Science, but it's a news piece, it's not a research article. So nitpicking on my part. And this is another example of that. Just so you know, this is this is in a scientific journal, Comparative and Functional Genomics, but this is a review paper. So this is published in a scientific literature, but it's not research literature. This is somebody writing about a bunch of other papers that have already been published. These are great for finding sources for things like what I'm going to keep asking you to do in class about write paragraphs about this and support it with research literature. Because these review papers are typically written with less jargon in them, typically. Sometimes they're not. So the problem here is that you could say, I'm citing this because this paper says the average DNA sequence differences is only 1.24%, and then we can talk about whether or not rounding is appropriate. But the actual citation is reference 7. So in this case, if you did this, what I'd like you to do is figure out at the bottom of this paper down in the literature cited section, what is reference 7, look that paper up. That should be the primary research literature that actually did the analysis and came up with that number. So in support, a lot of you cited this paper. Proofer et al., Proofer and others, Proofer and many other authors in Nature. And I'm just pointing out for a particular reason that this is from Svante Pobo's lab, because we're going to talk about his research group in just a second again, historical context. So this one says, what most of you said were this is 98% identical to corresponding sequences in the human genome. So if you round 98.7 up, you get to 99%. So a lot of you cited this as support of this fact. But then there were a number of you that found other great articles that I'd never seen before. Oh, thanks. So I did a lot of reading last night. And a lot of you pointed to this part. Let's see, where was it? More than 3% of the human genome is more closely related to either the bonobo or the chimpanzee genome than they are to each other. Some of you caught onto that 3% number and were, I think, arguing that that meant that we were at least 97% or less, 100 minus 3, 97% or less similar. But, and that's in this section right there. What they're arguing here is something a little bit different. They're saying that there are 3 different possible sort of family trees of humans, chimpanzees, and bonobos. One of them would be that chimpanzees and bonobos are closely related to each other, if you think about this as a family tree, and that humans are more distantly related. There are 2 other options. So what other pairs could be most closely related to each other instead of chimps and bonobos? Chimps could be most closely related to humans, with bonobos being the distant cousin. Or there's one other option. Chimps would be the distant cousin with humans and bonobos being most closely related. Those are the only three ways you can arrange those three groups to infer who's related to who. So what that 3% is saying is that that's what? Human more closely related to either the bonobo or the chimpanzee. So they're saying this relationship on top, chimps and bonobos being their closest relatives, is what they see up here. That's what happens when you look at 97% of the genome, you get that arrangement. And it's only 3% of the genome where you see something where humans look more closely related to one of the non-human primates. So they weren't using that to argue who is closely related to whom. They were just saying that there's 3% of the genome you can look at that doesn't tell us anything about who's related to who. Then some of you found this review article that says the difference between the two genomes is not 1%, but 4%. The difference here is critical, and that's what I'm going to end with, is what the difference is between how they did this study and that first article that said we were 99%. 97% of the time. Somebody found this fantastic, I couldn't download it because they wanted me to pay for $12 for it, so I didn't. But genetic differences between humans and chimps and among humans. Note that this is from 1979, though. So if you're going to take any other lesson away from this literature review for this question, it's that, and there's no right answer to this, should you put more emphasis on recent literature with modern techniques or older literature that might have been corrected since it was published? It's been a long time since 1979. I know because I've been around that long. And a lot has changed. So whether or not you trust this article, you could. It may have been true. I'm not saying either way. If this is true, it may have been true at the time, but maybe somebody has corrected this research. Some of you made a fantastic point. This is, again, a little bit of a side. I think some of you were a little bit anxious about whether or not we really are closely related to chimpanzees, which is fine. So a lot of the writing was sort of more negative, like, well, even if there's only 1%, this is absolutely a legitimate thing to say. Well, if we're 99% similar to chimpanzees, then that 1% that's different must be really critical because we're so great, which is absolutely legitimate. There is only a very small genetic difference between humans and chimps, but that 1% makes a huge difference. That makes us different from chimpanzees. And this citation that one of you found, or several of you found, says that. They said, yeah, there's only 1% difference between humans and chimps, but what does that 1% difference do? One of the things it does is it changes not just the sequence of the DNA, but some of those individual mutations that make us and chimps different cause changes in gene expression patterns. So it's not just that it changes the sequence of one protein, and that's it. It's that some of those really rare differences actually change the way and the number and the type of genes that are turned on in our brains. It has a huge effect. A small number of changes has a huge effect on development. On neurobiology, makes us humans and not chimpanzees, presumably. That was a great citation as well. The biggest difference in all of the papers that you sent me to read at midnight, those of you that did it at the end, there was a really critical difference. Are these different papers looking at what the quote is there, the fact, we share 99% of our genes with humans, bonobos, and chimpanzees. So you can imagine on a piece of chromosome, humans might have 3 different genes. That's human, chimp, looking at the same piece of the same chromosome, and those little right-angled arrows are the way geneticists indicate where transcription start sites are, the start sites of genes. So in the human, there's 3 genes. In chimp, there are 2. In bonobo, there are 2. So that means that in this region of the chromosome, we have both of the same types of genes. So we look like we share 100% of the genes in that part of that chromosome. But what about this gene on the left? It's unique to us. So if you looked at that whole thing, you would say, in terms of percent of genes that the 3 species share in common, what's the percentage?
67.
We share, of the 3 genes we have, 2 of them, 2/3, 67% we share. So that's one way to calculate difference between different organisms when you look at their genomes. How many genes, things that encode proteins, do we all have that are the same? That's different from percent of nucleotides in common, which is what all the genomics papers are based on, which is you sequence the genome, you get all the A's, T's, G's, and C's, and you ask how many of those are different. So that's like looking at one part of the chromosome in humans and chimps and bonobos and saying, hey, sequence alignment. Where do humans differ from chimps and bonobos?
At the second place.
Yep, number it, 1, 2, 3, 4, 5.
5th place.
We have an A and the two non-human primates have T's. And then there's also a difference there, but that's in bonobos, so we don't have to worry about that. So on average, in those 5 nucleotides, what's our percent similarity to chimpanzees? We've got 1 nucleotide out of 5 that are different, so that makes us look 80% identical. 80% of the nucleotides we share in common. So these are 2 totally different ways to measure, to get at the same question. So what a lot of you were saying when you answered this question, when you wrote your paragraph, was, well, this thing says it's 99% of our genes. That's this side. But what most of the papers you cited were talking about, where they were doing the whole genome sequencing. They weren't looking at genes. They were looking at, this is the sequence of the chromosome, line them up and count how many differences there are. Okay. That is a lot of detail we'll go through later in the course. So, with that said, let's vote. I'm going to open up one more Socrative activity, and then I'm going to tell you what I'd like you to do for next class, please. So I'm just going to ask you, now that I've summarized what a lot of you wrote, how do you feel about this fact? And whatever we decide will then become fact. You get the option of yes, no, or I don't know. Did I give you the option of saying I don't know? No. Okay, yes or no. Awesome. Black and white. So this is a short list of the references that you provided me, which was fantastic. Note they span from 2002 to 2012, which is fantastic. And so, 2 last things. Out of time. Right now in Google Classroom, you should also have a new practice. Some of you asked from last class to practice more of the sequence alignment, sliding the sequences left and right to line up motifs. So this is an exercise for 1 point, due Sunday night. It just got distributed on Google Classroom, Google Sheets. So please try that for next class. And then we're going to move on to the next topic. So not very much work. It looks like a long list. It's not going to take very long. And try, don't necessarily do or complete exercises 3.4 and 3.5. Okay. Have fantastic weekends, and I'll see you on Monday.